Okay, so we're we're in the home stretch here, and as I've listened to all these really great talks, I can't help but think that my talk is sort of a redux of everything we've been talking about so far in the past 2 days. So, you know, in my talk, there's going to be transposon sequencing, a little bit of type 6 secretion, and some colony resistance. So it's really sort of nice to have heard all of these talks. So my talk is entitled The Role of Shigella Sonnei Type 6 Secretion in Niche Occupancy. So a little bit of background on Shigella. Shigella is a leading cause of severe diarrhea with over 150 million infections globally each year. There are 4 disease-causing serovars. These are Shigella flexneri, Sonnei, Boydii, and dysenteriae. Shigella flexneri and Shigella Sonnei account for the vast majority of disease around the world, though. So something that's been appreciated for the past several decades now is that Shigella infections caused by Shigella flexneri are more associated with lower economic development. So areas of the world that have poor access to clean water, that's where you're going to find Shigella flexneri. Whereas Shigella Sonnei is more prevalent in industrialized nations. And so this is shown in this map of the world here, where it's plotting the ratio of Shigella flexneri to Shigella Sonnei, or Shigella Sonnei to flexneri. And you can see areas that are more darkly colored, typically areas where there's maybe considered more economic development, have greater ratios of Shigella Sonnei. Interestingly also, industrialization itself leads to a rapid shift from Shigella flexneri to Shigella Sonnei. And this perhaps can be best illustrated in China, which has in the past several decades undergone very rapid economic development. So even as recent as 2003, over 82% of the isolated Shigella strains were from flexneri, and only 17% were Shigella Sonnei. Less than a decade later, however, Shigella Sonnei has almost tripled and now accounts for almost 60% of the isolated cases, and Shigella flexneri is now only accounting for around 41%. And to make clear, the causes underpinning this changing Shigella landscape are not known. So what are the stages of a Shigella infection? So after ingestion of the bacteria and passage through the stomach, the first thing that Shigella will encounter is the barrier effect of the microbiota. Next, it will encounter the mucus layer, where Shigella will secrete mucinases, which allow it to slip through the mucus layer, where it then targets the colonic crypts. At this stage, it activates its type 3 secretion system and enters the tissue. Here in the tissue, it will spread and start to replicate, and this causes pro-inflammatory cytokines to be released, and a massive inflammation and neutrophil recruitment will occur, which comes to try and stop the infection. This eventually will cause the killing of the Shigella bacteria and tissue destruction, which basically completely destroys the epithelial layer, but will eventually lead to the resolution of the infection. So all of these stages have really been well documented and studied, and a lot is known about them, except for this very first stage. It's not been really appreciated what the effect of the microbiota is on Shigella infection. So the questions that I'm interested in the lab are exploring the mechanisms behind this Shigella flexneri to Sonnei transition. And to get at this question, I'm working on 3 different projects. The first is to look at the interaction of Shigella with the host microbiota, and we're developing a murine colonization model with Pascal von Esch in the lab. We're also doing a global overview of important genes in virulence and survival using TN sequencing libraries. And then together, these 2 projects have led me to study and enhance Sonnei competitiveness via a type 6 secretion system that it carries. So the bulk of the talk today will be about this middle point. So we started with the question, what genes contribute to Shigella Sonnei fitness in vivo? And we decided to use a TN-seq approach for this. So before we could go into that, we had to make sure that our Sonnei strain would be infectious, because there's a new model system developed in the lab looking at flexneri infection of the guinea pig. And the guinea pig is nice because it pretty much recreates all of the signs of disease and symptoms that occur in a human infection. whereas other model systems don't quite as accurately represent that. So we obtained a wild-type Shigella sonnei strain from the Pasteur strain collection. It was a clinical isolate from Paris in 1990. And when we infected guinea pigs with it, we saw that it was at least as infective and invasive as the lab Shigella flexneri strain. You can see on the right side the tissue damage that occurs when this strain is infected with guinea pigs. And you can actually see the colonic crypts being detached from the underlying epithelial layer. In this purple color, you can see, which is a stain for neutrophils, you can really tell that there are massive amounts of neutrophils just streaming into the tissue and all of the damage that goes along with that. So I won't go into too much detail about how we developed the TN library because we had a really nice talk that described that earlier. But suffice it to say that our pooled library contained over 168,000 unique insertions, which covered around 75% of the open reading frames that were predicted in Shigella sonnei. We took our pooled library and infected guinea pigs with 10 to the 10th CFUs, and after 8 hours, we were able to recover on average around 10 to the 7th CFUs. So we saw around 3 logs of selection occurring. Using a primer specific for the transposon that reads into the genome, and then sequencing the transposon genome interface, and then mapping it to our reference genome, we were able to find genes that were present in the control library but absent in the library that had been passaged through the guinea pigs. So when we started to look at our TN-seq data, we noticed at least one region that had several hits in close proximity to each other in genes that weren't readily identifiable. When we looked more closely, however, we discovered that they are genes that encode a type 6 secretion system. And so these are the genes that we hid in our screen are denoted by asterisks. And when we looked to see whether or not this genomic island would be present in Shigella flexneri, we found what appear to be the remnants of a type 6 secretion system. So this is in the same genomic location, but instead of having the full complement of genes, we only see around 6 remaining genes. And the type 6 system found in Sonnei is most similar to that found in pathogenic E. coli. So a little refresher on the type 6 system. Eric Cascal gave us a great talk yesterday about the structure and function, so I won't go too much into that, but I'll just remind everyone that type 6 systems are evolutionarily related to bacteriophages. They're used for niche competition and or virulence, and they're highly modular. So each bacteria can decide for itself what the full complement of its effectors are, and it sort of tailors that to what the bacteria needs to do. So type 6 systems are widely distributed amongst gram-negative bacteria, with some estimates ranging up to 25% of all bacterial species have a type 6 secretion system. That's of the gram-negatives. They require cell-to-cell contact to transfer effectors, and you can really see they create these very long, elongated structures in the cell. Okay, so when we started to try and test what the effect of the type 6 system might be doing, we very quickly realized that Shigella sonnei also encodes a colicin, and so the effect of which can be seen here. So if we are looking, so this is a liquid-based assay where we are refreshing E. coli. So if we take DH5 alpha, we see that after 90 minutes of refreshing, it starts to grow. If we spike the media with supernatant from Shigella flexneri, we see that there's no effect on the growth of E. coli. However, if we spike the supernatant with Shigella sonnei supernatant, we see a rapid decrease in the viable recovered E. coli CFUs. This also occurred if we took a type 6 secretion mutant. The supernatant from that also killed E. coli. So using these strains, we wanted to try a plate-based type 6 assay. And so what we did for this is we mixed the two competing strains together. So in this case, Shigella sonnei and E. coli. So if E. coli is left alone, it grows to high levels. However, if we then compete Shigella sonnei in, we see a massive amount of killing occurring. If instead of competing with the wild type, we compete with a type 6 mutant, we see a dramatic decrease, or we see around 3 logs increase in recovered E. coli CFUs, and we can complement this phenotype. So this is using colicin-sensitive E. coli. So we wanted to then sort of strip out the effect of the colicin, so we isolated a colicin-resistant strain of E. coli and did the same plate-based competition assay. And in this case, we can narrow down the effect of the type 6 system. So when grown in competition with Shigella sonnei, we see fewer CFUs recovered than when we compete with the type 6 mutant. We can also visualize this because the E. coli strain we're using has a DS-red fluorescent plasmid. So when it's grown in competition with Shigella sonnei, the colony becomes white, whereas if it's grown with the type 6 mutant, the colony is red. We also tried competing against Shigella flexneri 5A or a more clinically relevant 2A strain, and we saw no competition with E. coli using Shigella flexneri. So then we asked the question, can Shigella sonnei compete against its relative Shigella flexneri? And what we saw that also occurred. So if we put Shigella sonnei and flexneri together, we see on average around 4 log CFUs, whereas if we put it with the type 6 mutant, we get closer to 6 logs recovered. And again, you can see the difference in color of the colonies. So this competition required contact, as growth in liquid did not yield any differences between the wild type and the type 6 mutant. So then we decided to go in vivo. So for this experiment, we chose the mouse. And the reason we chose the mouse was because mice do not normally get the symptoms of Shigella infection. They don't have the massive neutrophil inflammation and tissue destruction. So this allows us to really narrow in and focus on the interaction with the microbiota. So if we infect mice with just Shigella sonnei, in the tissue, we see after 24 hours around 5 logs of CFUs. If instead we infect with the type 6 mutant, we see very few CFUs in the tissue after 24 hours. And if we pre-treat the mice with streptomycin to lower the microbiota, we now see much higher CFUs in the tissue, as well as we no longer have a phenotype with the type 6 system. So it seems that the removing the microbiota allows Shigella sonnei, whether or not it has a type 6 system, to enter the tissue. We also did this experiment with Shigella flexneri, and Shigella flexneri acted very similarly to the type 6 mutant of sonnei. All of these phenotypes were similar whether we looked at the tissue or the feces, and at 24 and 48 hours. It's the same trend in either case. Sonnei wild type always seems to do better than the type 6 mutant, and that usually can be removed by streptomycin treatment. So looking at the histology of streptomycin pre-treated mice, we see that Shigella sonnei, whether or not it has the type 6 system, is able to enter the tissue, but it does not cause the tissue destruction that we see in the guinea pig. So if you remember the guinea pig, it was all torn up. Well, this is much smoother. It's fully intact, but the bacteria are still in the tissue. So finally, we wanted to see whether or not Shigella sonnei would compete with another bacteria in vivo. So we pre-treated our mice with streptomycin, and then we simultaneously infected them with both Shigella sonnei and Shigella flexneri. And after 48 hours, we recovered the bacteria from the colon. And we're looking here at the numbers of Shigella flexneri that we had. So if we put the wild type sonnei and flexneri together, we see around 1.5 log CFUs after 48 hours. However, if the Shigella flexneri was competing against the type 6 sonnei mutant, now we see around 3.5 to 4 log CFUs in the tissue. The same occurred in the feces as well. It's the same story there. So then if we look in the histopathology, we see that it's a little hard to see, the green's not coming through, but when type 6 mutant is competed with wild type Shigella flexneri, there are Shigella flexneri and Shigella sonnei cohabitating happily, which we don't really see in the wild-type sonnei. So our working model for now is that the wild-type Shigella sonnei uses its type 6 secretion system and collicin to potentially clear a way for the bacteria to contact the mucus, and then potentially a second time when it might need a type 6 system is in the crypt, where previous work in the lab has shown that there's a crypt-specific microbiota, which may also compete with Shigella sonnei. On the other hand, if we have a type 6 deletion mutant, or if the bacteria has lost its type 6 system, as has Shigella flexneri, it is likely impaired in its ability to access the mucus layer and may also have a blockage in the crypt. So just by way of perspectives and conclusions, we ran a TN-seq screen that identified type 6 secretion system in Shigella sonnei during our guinea pig infection. In vitro, Shigella sonnei antagonizes closely related E. coli and Shigella flexneri strains using its type 6 system. The type 6 system is required for survival and competition during challenge in a mouse. And this raises the questions that we're currently trying to answer is, does Shigella sonnei target specific competitors in vivo? I think it's fairly likely that there is one or two bacteria that it really needs to clear out before it can colonize. And then sort of the more larger question, can the survival advantage conferred by the type 6 system help to potentially explain some of the transition towards Shigella sonnei? So with that, I'd like to acknowledge my Philippe Sansonetti, Benoit Martin, Pascal Vonesh, and Azadeh Safarian, who have all contributed immensely to this work. And I'll take any questions.